Here we go. So, well, function, know that, like, regulation, endocrine glands produce hormones, they go in the blood. And that actually goes into the question number 2, exocrine in the, lumens, endocrine glands produce hormones that go in the blood or extracellular fluid. Know your primary endocrine organs, know what they are. They produce hormones. Secondary, I'm not going to ask you the list, but if I ask you whether something is secondary or primary, you tell me. Clear? 2 systems, nervous and endocrine. The table that we have, fairly, it's pretty simple. Like endocrine, for instance, endocrine effects last longer, but takes more to develop. Stuff like that. Okay? Like, which of the following statements is correct? 2 main types of hormones. Water lipid soluble. In terms of chemical functional subtypes, what I expect you to know is amino hormones, peptides and proteins for water soluble, and thyroid hormones, eicosanoids and steroids for lipid soluble. Receptors for lipid soluble inside the cell because they can cross the membrane. This complex, look, I will ask you, there will be several questions on this. Complex, not the hormone, binds to the DNA, initiates transcription, which leads to RNA, which then is translated into a protein. Okay? Know this. I will ask this. Water-soluble hormones, receptors on the surface, kind of signaling, second messenger signaling. CAMP, binding to the receptor, activation of G protein, activation of adenylate cyclase, synthesis of cyclic AMP. Remember what I told you in the lecture, it's not going to go any farther. It will not. Okay? Ip3 interaction, G protein, phospholipase C, it produces Ip3. Ip3 leads to the release of calcium from endoplasmic reticulum. It's not going any further. Receptor-bound tyrosine kinases, tyrosine kinase phosphorylates the protein. Before moving on, anything that is unclear. If something is unclear, stop me right away. Okay, we good? 3 stimuli: humoral, nervous, hormonal. There will be a question. I give you examples. You tell me which of the following is humoral stimulus, which of the following is nervous. Does that make sense? You read the description, tell me this is like insulin in response to glucose. That's your humoral. Got it? We all clear? I've got a question recently. What type of change does humoral stimulus reflect? Change in the composition of extracellular fluid, change in basically the bodily functions. You can consider muscle, I don't know, like pressure being, no, pressure is not humoral, that's a nervous stimulus. For oxytocin, that's a nervous. Because signal goes from cervix to the brain. But like insulin stimulation, that's humoral. Does that make sense? Aldosterone release, response to humoral. Nervous, duh, you'll understand. Nervous stimulus. Hormonal, in any axis, like growth hormone, can be stimulated by the hormonal stimulus. We clear? Location. Autocrine, paracrine, and endocrine actions. Action on the same cell is autocrine, adjacent paracrine, distant endocrine. Negative feedback, Now look, I'm not going to ask you the question what type of feedback, but it's a question for you, so you keep it in mind, and you apply the knowledge of negative feedback to your answers. You can check, did I answer this correct? Does that represent negative feedback? Am I clear? For radioimmune assay, I'm tempted to go over it again, but I won't, because there's going to be 1 or 2 questions regarding analysis of hormones. And what you really need to distinguish, if you see the word radioactivity, we talk about radioimmune assay, and vice versa. You have to know what's radioactive, right? If I ask you about radioimmune assay, you have to know that hormone in the standard is radioactive, in the sample it's not. Okay, and they compete for antibodies. If we talk about ELISA, it's the complex between the hormone and the antibody that is detected by another antibody. The purpose of this part of study guide, when you answer those questions, you get a better, clearer picture of what these methods are. And when I ask you a question with a bunch of statements, you can better correlate these statements to each of these 2 methods. Am I clear? Good? Awesome. This is, yeah, just know that you're going to get a question. Which of the following is hypothalamic hormone? Or not? We good? Answer to this question. Neurons. What do they have in common in terms of regulation? Hypothalamic hormones either stimulate pituitary or stored in there. The following several thingies, okay, they all refer to different hormones. Now look at this. If you would try to answer it when we just got to hypothalamic hormones, it wouldn't make any sense to you. How do you know what is the stimulus for the release of thyrotropin releasing hormone, or TRH? Can you answer this question now? What is the stimulus for the first hormone of hypothalamus pituitary thyroid axis? Huh? Cannot hear you. No, for thyroid. What is the effect of thyroid hormone? Increased BMR. So what's the stimulus for the whole axis? Low BMR. Does that make sense? Apply your knowledge about the whole regulation thing. Because when we say that thyroid hormone is stimulated by the low BMR, it's not direct. Low basal metabolic rate stimulates TRH, TRH stimulates TSH, TSH stimulates thyroid hormone. Does that make sense? What I'm essentially asking you here is what these axes start with. Does that make sense? Do you understand? Like, let's play one more. What are the stimuli for the release of GHRH? Can you think of? What is the ultimate result? When GHRH is released, what is the ultimate result at the end? Growth hormone. What is it? What are the stimuli that we talked about? Exercise, sleep, okay, decreased blood glucose, decreased blood lipids. Does that make sense? All of this will initiate the whole signaling. Am I clear?
So, whenever it says, what are their functions?
What do they do? Like, what does GHRH do?
Oh, okay. Yeah.
Leads to the release of growth hormone. What does TRH do? Releases stimulates the release of TSH. Make sense? Good. For prolactin inhibiting, prolactin stimulating, you only need to know the function. So they stimulate prolactin, which stimulate milk production. Where is pituitary gland? In sella turcica, under hypothalamus. Infundibulum touches it. 2 parts. I'm not asking about adeno and neurohypophysis. I'm asking about anterior and posterior pituitary glands. Which another endocrine gland here? Hypothalamus. Vascular system that connects. Remember, hypothalamus, pituitary, portal system. Main parts: primary plexus, portal veins, secondary plexus. So this is something that you need to know for anatomy. By the way, somebody asked me about the study guide for anatomy. Shoot me an email. I keep forgetting things like that. Just shoot me an email saying, I had really annoying kid in another EP2 class who kind of overwhelmed me with emails. Are you going to post the study guide for anatomy? Are you going to post the study guide for anatomy? She said, screw you, I'm going to post the study guide for anatomy. So, annoy me. When I say this, delivery step by step, hypothalamic neuroendocrine cells release hypothalamic hormones in the primary plexus. They move with blood from the primary plexus through the portal veins into the secondary plexus and act on anterior pituitary. Does that make sense? They're all listed in the table. We clear? When I say metabolic, I mean levels. So low glucose, low fatty acid will stimulate the release. Elevated amino acids will stimulate the release. Am I clear? Non-metabolic stimuli: exercise, trauma, sleep. Those 3. We clear? When I ask you this, hypothalamus produces GHRH, which leads to the release of growth hormone, right? Which then has 2 types of effects. So you need to know direct and indirect. And remember that indirect are mediated by insulin-like growth factors. I think it's a lab number 2. You had to draw this regulatory flow chart. Be comfortable with that. Does that make sense? Elevated glucose, elevated blood lipids. When I say elevated, it's in blood. That's it. That's it. Okay. Because look, Natalie. This is what stimulates growth hormone, right? And this, quite opposite, are effects. That makes sense? Effects, in general, are opposite to the stimulus. No, it's fine. That's it. Indirect, there are many more. Protein synthesis, growth of bones, muscle growth. Yeah, that's true. But those are mediated in between by IGFs. So we just talked about this, indirect effects. Here, I refer to the negative feedback. So remember, the final product will inhibit the original hormones, right? So IGF will inhibit the production of this and this, but will stimulate the release of growth hormone inhibiting hormone from hypothalamus. Any questions so far? Metabolic stimuli that inhibit. So, let me ask you this. If growth hormone increases blood glucose and blood lipids, if its action is to increase those 2, what are the metabolic stimuli that will inhibit growth hormone? It's a simple question. Increased blood glucose and increased blood lipids. Does that make sense? Okay. Non-metabolic stimuli, I refer to age and obesity. Okay. May I skip this too? It seems super simple, right? You know gigantism, dwarfism, acromegaly. You will be able to tell them apart, will you? I don't have to worry about it. That was a question. So, thyroid. Inferior to larynx, anterior to trachea, main anatomical parts that you need to know, 2 lobes, and isthmus. Microanatomy. I don't require to tell parafollicular and follicular cells apart, okay? But if you have a question and you have a picture of thyroid gland, don't tell me that those are chief cells or oxyphil cells or beta cells, okay? There's going to be only one answer that logically falls into thyroid gland story. Follicles filled with colloid are building blocks. 2 types of cells, follicular, parafollicular. Follicular produce thyroid hormone, parafollicular calcitonin. Calcitonin is water soluble. Thyroid hormone is lipid soluble. Amino acid required for the synthesis. Tyrosine, necessary chemical element iodine. Look, this is all memorization. Step-by-step process. To what degree of depth do you have to know it? Iodine goes from the blood through the follicular cell all the way into the follicle where it modifies thyroglobulin, specifically tyrosine residue. Does that make sense? Blood, follicular cell, follicle. In the follicle, it modifies the protein, thyroglobulin. Not just the whole protein, but tyrosine residues. Essentially, at some point, you have this... Jenna, real good? Thyroid hormones hanging off the thyroglobulins. Thyroglobulin ends up in the lysosome. Thyroid hormone is cleaved off, released in the blood. That's the depth of knowledge. I'm not asking any chemistry whatsoever. Stimuli that increase production. Low BMR. Physiological effects. Now, high BMR is not a good answer. Remember we discussed things like increased cardiac output or increased heart rate or increased respiration, perspiration, stuff like that. Protein synthesis, breakdown of fat. You have to understand what happens when BMR increases. Are we clear about it? Okay? So I'm not going to ask you, you're not going to get an answer just elevated BMR. If I ask you, you need to, out of the list, you pick, say, elevated heart rate. Does that make sense? HPT. That's our standard axis. Hypothalamus, pituitary, some kind of a gland. Does that make sense? If it's TRH, TSH, thyroid, that's hypothalamus, pituitary, thyroid axis. If it's CRH, ACTH, cortisol, hypothalamus, pituitary, adrenal. Gonadotropin releasing, FSH and LH, testosterone, hypothalamus, pituitary, gonadal. Am I clear? Please work on this and use the approach known as brain dump when you get to the exam. Okay. This question, if TH levels go up, these two go down, and vice versa. Receptor inside, it's fat soluble. Look, this is again complex binds the DNA, stimulates the transcription, translation, whatnot. Dietary goiter, no iodine, no TH, extremely high levels of TSH. It stimulates growth of thyroid gland, F goiter. Does that make sense? So you have to, there will be questions about those diseases. Treated, cured, get more iodine. Why thyroid gland increases in size? It compensates. Graves' disease. Antibodies bind to which gland? Thyroid. Increased production of TH. So you will see a lot of TH, little TSH, little TRH. Remember, please, that antibodies bind to thyroid, not any other. Physiological effects, hyperthyroidism, you're really hypo, you're lethargic. Myxedema, complete lack of thyroid hormone, swollen face because of the interaction between the hyaluronic acid and proteins in the subcutaneous interstitial space. Hashimoto disease, hypothyroidism. Antibodies this time inhibit the release of thyroid hormone. So in Hashimoto's, TH levels are low. What about TRH and TSH? High. Yeah, that's a repetition. Overlooked it. Parathyroid gland. What you need to know about parathyroid, where that is located. Stimulus. Low calcium. Effects of PTH. Elevated calcium. Now, I want you to know 3 mechanisms how blood calcium goes up. One is stimulation of osteoclasts and bone resorption. 2, calcium is not excreted by the kidneys and taken back. 3, PTH activates vitamin D, and vitamin D increases calcium absorption in the intestines. Am I clear? PTH activates vitamin D, which stimulates calcium absorption in the intestines. Calcitonin, produced by parafollicular cells of thyroid gland. Stimulus, extremely high calcium in the blood. Physiological effects. Calcitonin stimulates osteoblasts, and they deposit calcium in the bones, meaning that calcium levels in the blood will go. What is the stimulus for calcitonin? High calcium. What is the physiological effect? Go down. Make sense? Hypercalcemia. Muscle weakness, kidney stones. Hypocalcemia. Overactivation of muscles and nervous system, tetanic muscle contraction. Adrenal, cortex, medulla, you do have to know that. You kind of got a gist when we discussed the quiz today. Medulla, water soluble. Cortex, lipid soluble. 3 layers, zona glomerulosa, zona fasciculata, zona reticularis. I reserve the right now. We'll ask, cortisol is produced in, tell me, zona fasciculata, okay? I can do that. So that's this. You need to know which zone produces which hormones. So for aldosterone, stimuli, we have 3, right? One stimulus is low sodium in the ECF. That leads to increased sodium retention. Make sense? And, followed by the increased retention of water. Stimulus number 2, low blood pressure, which stimulates Renin-Angiotensin-Aldosterone System, and increased aldosterone levels will keep sodium in, which will be followed by water going in, which will lead to elevated blood volume and blood pressure. Are we clear? Sodium and water retention. Now, third stimulus is potassium. Remember, elevated potassium leads to increased aldosterone production, and aldosterone will increase potassium excretion. We good? So that's reiteration of what we said before. If you inhibit aldosterone, what's going to happen to the amount of urine that you produce? Aldosterone retains sodium and water. So if you inhibit, yes, you will produce more urine. You will also excrete sodium with urine. That makes sense? So sodium, not ECG, ECF. Why ECG? It's not cardiogram. Excess aldosterone. If you have too much aldosterone, what's going to be happening to the volume of your urine? It's going to be really small. Your blood pressure will go up, and your sodium amount in the body will go up. Potassium will be excreted. Excessive aldosterone, how will it affect performance of the heart? What do you think? It's kind of a tricky question. What if you have a lot of aldosterone? What you're going to excrete? Potassium. Is potassium important for the heart function? Yeah. So you're going to have hypokalemia. Okay. Receptor is inside the cell because aldosterone is lipid soluble. Signaling system. Now you have the full picture. Renin-Angiotensin-Aldosterone System. What activates this system? Anyone remembers what activates renin? Low BP, low blood pressure. Now, so for ANP, for atrial natriuretic peptide, what is the stimulus for ANP? High blood pressure, right? If aldosterone and atrial natriuretic peptide are opposite, aldosterone is stimulated by low blood pressure, ANP is stimulated by high blood pressure. Okay? ANP inhibits aldosterone production, sodium becomes excreted, water follows the sodium, so you pee a lot, remove a lot of sodium from your body, your blood pressure falls. Cortisol, remember it's stress. Now, look, I'm not gonna talk about that same concept, the axis again. You get the gist. Am I correct? You understand what I'm talking about. So if I tell you, if you will start shooting someone with cortisol, how is it going to affect the levels of CRH and ACTH? Tell me. Exogenous cortisol is going to reduce ACTH and CRH because of the negative feedback. Are we clear? You have to know this axis. Metabolism. Blood glucose goes up. Lipids, I'm going to write lipolysis. And proteins get broken down. It also suppresses your immune system, reducing inflammation. Metabolic condition, diabetes can develop as the result of chronic stress. Why is stress often accompanied by elevated blood pressure? Because along with cortisol, aldosterone is stimulated. It will increase sodium retention, water retention, and will elevate blood volume and blood pressure. Am I clear? Receptor for cortisol, again, fat soluble inside the cell. High levels, Cushing's, low levels, Addison's. Now, I can ask you a question in Cushing's. How do you expect CRH and ACTH levels to change? You think, okay, Cushing's, very high cortisol, CRH and ACTH are low. Okay. Now, androgens from zona reticularis contribute to adrenogenical syndrome. You answered it today brilliantly. Okay. No glucocorticoids, a lot of ACTH. A lot of ACTH overstimulates adrenal cortex. It produces a lot of androgen. masculinization in females. In males, it's going to be the opposite. A lot of female androgens, which will lead to development of ambiguous reproductive organs. Posterior pituitary, 2 hormones, oxytocin, ADH. Now, remember, I can ask you, how does, hypothalamus communicates with posterior pituitary? You tell me, hypothalamus delivers hypothalamic hormones directly into posterior pituitary by the means of hypothalamo-hypophyseal tract, which consists of neurons. Oxytocin, know the positive feedback for it. I will ask about this. ADH, questions like the one that we worked out here. So, if you are dehydrated, your osmolality of extracellular fluid goes up. That stimulates release of antidiuretic hormone, which leads to retention of water in kidneys, which leads to increased blood volume, increased blood pressure, normal concentrations of solutes in your ECF. In the meantime, when ADH is released, volume of your urine will become smaller, right? Does that make sense? And it becomes really concentrated because kidneys suck all the water out of the urine. Okay? For decreased, it's going to be absolutely opposite. Effect of ADH inhibitors. It's diuretic. You start peeing. Diabetes insipidus is the deficit in ADH production, which leads to constant thirst. Anatomical location. 3 anatomical parts. Never mind. Yeah, I think never. If there's, I don't remember if there's going to be a question. Never mind. It's a head, tail, and a body. Okay. If this is the pancreas, and this is spleen, this is head, tail, body. Structures in the pancreas. Langerhans islets. Cells that you can find there. Alpha, beta, delta, and PP cells. You need to know the functions of alpha, beta, and delta. That is it. Okay? Make sense? So, delta cells, you've got somatostatins for beta cells. You've got insulin, metabolic effects. So this is something that I would love to go over today, if we have time in the lab. I'll give you the lab, you'll keep it. Remember, insulin stimulates glycolysis, glycogenesis, lipogenesis. Does that make sense? And glycolysis, glycogenesis, lipogenesis. Glucose uptake. Anything that removes the glucose from the bloodstream. What it inhibits? Gluconeogenesis, glycogenolysis, and lipolysis. Did I say lipogenesis? I think it did. Okay, lipolysis, gluconeogenesis, glycogenolysis. Anything that breaks down your storage to release glucose is inhibited by insulin. Does that make sense? Those questions, I'll be honest, they're important, but they represent maybe the most difficult questions for you. Make sure that you know the terminology and you understand what insulin does. This is the scheme how glucose stimulates insulin release. Okay, remember, glucose goes in the cell, leads to the increased levels of ATP. ATP close potassium channels. Potassium stays in the cell. Beta cell is depolarized. It is depolarized. Calcium channels open, and calcium leads to the release of vesicles containing insulin from beta cells. So the more glucose, more ATP, more potassium channels closed, depolarization, more calcium, more insulin. Insulin interacts with its surface receptor, which carries tyrosine kinase, which phosphorylates a bunch of proteins. Glucose uptake reduces the glucose levels in the blood. People that don't have insulin switch to fat as the source, right? So fat metabolism leads to accumulation of keto acids. Keto acids leads to acidification of the blood. Acidification of the blood may lead to coma. Why do athletes use growth hormone and insulin together? Insulin requires uptake. Insulin leads to uptake of glucose and amino acids. And growth hormone stimulates the protein synthesis. Does that make sense? Either of them is not effective. Does that make sense? No? Absolutely. So insulin is needed to stimulate uptake of amino acids and glucose. And growth hormone stimulates the protein synthesis, the use of amino acids. If you have only growth hormone, amino acids stay in the blood. They don't get into, say, muscle cells. Make sense? If you have only insulin, amino acids are taken into the muscle cells, but there is no growth. Are we clear? They are, both are required for efficient growth. Okay. Type 1 diabetes, lack of insulin. So in patients with type 1, insulin levels are low, glucose levels are high. Treatment, insulin. Type 2, insulin resistance. So insulin levels are normal or high. Treatment is lifestyle changes. Comorbidities associated with type 2. Obesity, metabolic syndrome, hypercholesterolemia, all those lifestyle things. Glucagon alpha cells. Glucagon is a protein. Metabolic stimuli, low blood glucose. Metabolic effects of glucagon. Glucagon stimulates lipolysis, glycogenolysis, and gluconeogenesis. Does that make sense? So, insulin and glucagon stimulate opposite processes. Glucagon receptors on the surface, it stimulates second messenger activation. Male gonads, testes, HPG axis. Now, I want to be very clear about it. The only questions about the hormonal regulation, I will ask about the male HPG axis. So, if I ask you about the effects of testosterone on LH, FSH, gonadotropin releasing hormone, testosterone inhibits it. Am I clear? That makes sense? So, gonadotropins stimulate the release of testosterone, and that's it. We're good? Testosterone is necessary for maturation of sperm. Remember that. Negative feedback, what we just said. Testosterone inhibits gonadotropin releasing hormone, FSH, and LH. Effects as a whole: promotes growth, makes skin oily, deepens the voice, leads to growth of skeletal system and secondary sexual characteristics. Female gonadal hormones: estrogen, different types of estrogen. Okay. Now, I'm not going to ask you about HPG axis in females. Deal? Like all this positive feedback between estrogen and luteinizing hormone, don't worry about it. I will ask you about the effects. Like ask you what are the functions of estrogen? What does it do? Okay? It leads to like secondary sex characteristics development. It is necessary for proper follicle maturation. That makes sense? You understand what I'm trying to say? But I'm not going to ask you about that endocrine axis yet. When we get to reproductive, huh?
But you want us to know the male axis.
Male axis, absolutely, yes. Effects, I want you to know the effects. Like if I ask you what are the effects of estrogen on skeletal system? Calcium storage, proper calcium storage. No. Just no. Don't worry. Not going to ask this. Pineal gland, melatonin, sleep cycle. Precursors is tryptophan and serotonin. In the daylight, melatonin is not produced. During nighttime, melatonin is produced. Thymus, thymopoietin, thymosin, and thymolin, 3 hormones that regulate maturation of T cells. Thymus activity decreases during lifetime. Now, secondary. ANP, already discussed. So for these hormones, you have to know the stimulus. Okay. Stimulus here, high blood pressure. Effects, natriuresis, release of sodium, and lowered blood pressure. And it's also a vasodilator. It inhibits Renin-Angiotensin System. Ghrelin, hormone of hunger, stimulus lack of food. Histamine and gastrin, stimuli presence of food, right? Effects of ghrelin is similar to glucagon. It actually stimulates glucagon release and increase blood glucose temporarily. Gastrin and histamine. Gastrin stimulates histamine. Histamine stimulates release of hydrochloric acid. This for intestines. Stimulus, food in the intestine. Secretin, bicarbonate release. Cholecystokinin, release of enzymes from the pancreas. Effects, inhibition of gastric activity and increased release of insulin. Renin is in the kidneys. Stimulus that stimulates renin is low blood pressure. Renin stimulates angiotensin. Angiotensin stimulates aldosterone. Aldosterone stimulates sodium retention, water retention, and elevates blood pressure. Erythropoietin produced in kidneys, and it stimulates more red blood cells. Cool. Osteocalcin produced in the bones. The main effect that I want you to know for osteocalcin, and I don't ask about the stimulus because it's kind of unknown. Main effect of osteocalcin, elevated insulin levels. Somatomedin or IGF produced by liver, as well as angiotensinogen. For somatomedin, the stimulus is growth hormone, okay? And metabolic affects growth. Insulin-like growth factors stimulate growth. Endocrine effects, somatomedin inhibits growth hormone, releasing hormone, and growth hormone itself. So basically this story harkens back to the growth hormone regulation. Stimulus for conversion of angiotensinogen to angiotensin is renin. The downstream effects of angiotensin 1, angiotensin 2, and aldosterone. Thrombopoietin is produced by liver, stimulated by low levels of platelets, effect high levels of platelets. Adipose tissue produces leptin. Stimulus is satiety. Effects makes you feel full. Okay. Deficit of leptin leads to obesity. Ghrelin exhibits effects opposite to leptin. Adipose tissue produces adiponectin. Main functions: anti-inflammatory, cardioprotective, regulation of the food intake. Cardioprotective, anti-inflammatory, improves insulin sensitivity. In obesity, adiponectin levels are chronically down and leptin levels are up. Before we wrap it up, any questions? Very specific questions on the study guide. If not, then